Dear students, welcome to my channel. In this video lecture, we'll learn gel filtration chromatography. Let's start. Gel filtration chromatography, also known as size-exclusion chromatography, is a technique used to separate molecules based on their size and shape. This method utilizes porous gel particles as the stationary phase, which function as sieves to separate molecules based on their ability to enter the pores. Smaller molecules can penetrate the pores and are retained longer with the column, while the larger molecules are excluded and eluted faster. The size-based separation allows for the isolation and purification of different components from a mixture. So in gel filtration chromatography, we use gel as the stationary phase, and the separation mechanism is the smaller particles are retained on the pore of the gel, but the larger particle just bypass the gel pore. Okay, so larger particle elute first and the smaller particle or smaller molecule come later. So this is the some synonyms of gel filtration chromatography. So first one is size exclusion chromatography. As the name suggests, size exclusion, that means the particles or the molecules are separated based on their size. Okay, then it is also called molecular exclusion chromatography because molecules are getting separated and excluded in accordance with their uh, size and shape. Then it is also called molecular sieve chromatography or MSC because in this case sieving is being performed. Okay, particles are getting separated based on their size. So the term the sieve, we actually are introduced with this term uh, from the early times because we have read this in pharmaceutical technology during the dry granulation or wet granulation method. We conduct this sieving. Okay, so the next one is gel permeation chromatography because the molecules or the proteins or the DNA, the samples are permeating throughout the gel. And the last one is gel chromatography. So here at the stationary phase, this is the column. Okay, this is the mobile phase are getting eluted. Samples are getting eluted based on their size. So you can see the gel bits and also can see the inner structure of the gel bit. We can see here the gel bit contains some pores. Okay, small particles as we can see are traveling through the pores. Okay, because their size is smaller, the particles are also smaller, so they are coming through this, but the bigger particle cannot pass throughout these pores. They just come beside the pores. Okay, so they get eluted faster, but the smaller particle are retained and take longer time for their separation. Hence the larger particles are eluted first and the smaller particle eluted last. So we can see here the size of the protein. So there are three proteins that we should separate it. We can see the stationary phase, the gel bits. So whenever we are applying the sample, smaller particles or molecules are retained on the pores, but the larger particle are eluted first, and in the chromatogram we are having a peak. Then the medium-sized particle are getting out, we are getting a peak, and finally the smaller molecule are getting out, and we are getting the peak. So in the chromatogram or in the PC, we will just see the absorbance. Okay, we'll see the area, peak area or the peak. So you have to analyze your sample based on these peaks. Okay, because you will not see the molecule or you will not see the separation. You will only see the SPLC or GFC peaks. Okay, you have to analyze your sample, you have to identify your sample, you have to separate your sample, and you have to quantify your sample based on the peak. Okay, so let's see the principle of gel filtration chromatography. It separates molecules based on their size and shape. The stationary phase consists of porous gel particles that act as sieves, allowing smaller molecules to penetrate and to be retained longer, while the larger molecules are excluded and elute fast. So you can see here instrumentation of GFC. Here is the mobile phase or the buffer media. This is the recorder. Here sample are applied. This is the sample preparation tray, and this is the recorder. Okay, so these are the overall uh, instrumentation for GFC. So gel filtration relies on the differential penetration of molecules into a porous matrix. The matrix, typically composed of crossed-link polymer like dextran, agarose, or polyacrylamide, contains pores of various size. So stationary phase contain the pores. The larger molecule cannot enter the pores of the matrix and thus elute quickly following the mobile phase. So mobile phase is usually a buffer. So why buffer? Because in gel filtration chromatography, we analyze protein, DNA, RNA, polymer. Okay, so these are very sensitive component. So they usually requires buffer and they cannot tolerate highly acidic or basic media. Smaller molecules can enter the pores and are delayed because they must navigate through the internal spaces within the beads, resulting in a longer path and thus eluting later. So here we can see the particles are getting separated based on their peaks, say size, and we are having the chromatogram. Okay, so this is a chromatogram. When some compounds are eluted, then a chromatogram appears in the recorder or in the computer. When some compounds are eluted, then we are getting a peak. So by analyzing the peak, you must need to understand the nature of the protein, the quantity of the protein, the amount of the protein, uh, the concentration of the protein, okay, or the presence of any impurities in your sample. Because if you have any impurities, then several other peaks could appear here. Okay, so this is the overall approach for determining the protein's concentration or quantity in the sample. So basic principle, as we have already discussed, here we can see whenever we are applying the sample, then they are getting separated. Okay, we can see protein A, B, C. So from here we can easily understand that the particle size of A is very much smaller. The particle size of the molecular size of C is very much larger. Okay, so another view has been shown here. You can easily understand that larger molecule are just bypassing the pores, but the smaller molecule are getting retained and getting throughout the pores. Okay, so they are taking the higher time. So let's see the steps of gel filtration. First of all, we need to do column preparation. So a gel filtration column is packed with porous bead and the column is equilibrated with a suitable buffer. Okay, this is as like as the column chromatography. Then we have to apply the sample. A sample containing a mixture of molecule of different sizes is applied to the top of the column. Then elution. The sample moves through the column as the buffer flows, separating based on molecular size. Large molecule elute first, followed by intermediate, and then small molecules. After that, the detection and collection. As the different fractions elute from the column, they are collected and analyzed, typically using UV absorbance or other detection method. Okay, so these are the overall step-by-step procedure for gel filtration chromatography. So we can easily understand that our stationary phase is playing a vital role in the separation, because it is not as like as the other stationary phase like column chromatography or TLC, okay, or SPLC. It's not like that. It is having some property like it's containing pore and retaining the smaller particles. Okay, so moreover, our stationary phase should be very much pure, non-irritant, non-toxic, and very cost-effective. And it should not be as like as the stationary phase used in column chromatography or any other chromatography, because In that chromatographic technique, we are actually separating the proteins. Our sample are proteins or DNA, RNA, which are very sensitive and very selective. Okay, so we can say here we use this stationary phase like agarose based gel like saffrose, then dextran based gel like Cefadex, polyacrylamide based gel like biogel P. Okay, composite gel like dextran agarose like Superdex or Superrose. Okay, we also use silica based gel. So we use this for small or large molecule separation and analysis or isolation. So let's see the basic components of GFC. As we have already discussed, it is composed of stationary phase. Here are the stationary phase, then mobile phase. The buffer is the mobile phase. The column, this is the column. The pump. Why pump is used? Pump is basically used for pressure generation. Okay, to analyze the sample quickly. And lastly, detector. We can see here a detector. So let's discuss one by one. So stationary phase. It is composed of semi-permeable porous polymer gel bits with defined range of pore size. So they should be chemically inert, mechanically stable, ideal and homogeneous porous structure, uniform particle and pore size. Pore size of the gel must be carefully controlled. So then the mobile phase. An organic solvent is used as mobile phase. Buffer is used to maintain the pH. So why buffer? Actually, we would see that in our protein lecture, we have discussed that protein maintains a isoelectric point. Okay, you know, in an isoelectric point, protein remains in as deuterium. Okay, whenever you are changing the pH, then its nature will ultimately get changed. Even by changing the pH, also your protein may get denatured. So it is very critical to maintain a proper bufferized condition in the protein sample. Okay, because protein is structure is very sensitive. You know, it has a 3D structure. You know, protein structure is three-dimensional like that. Protein has a like that structure, and these are the amino acid residues. There are some alpha helix and beta sheet. Whenever you are changing the pH, the protein may get denatured and may get destroyed. Okay, so we have to carefully monitor this buffer system. The samples are eluted isochronically, and there is no need to use different buffer during the separation. Then column, there are two types of columns like analytical column and preparative column. By analytical column, you usually quantify and we analyze or we undergo the assay of proteins. And by preparative column, we undergo isolation, separation, or purification of proteins. So you can easily understand that analytical column are much more sensitive than preparative column. Because by analytical column, we are determining the concentration. Okay, but by preparative column, we are just isolating or separating the protein. Then the pump, they are either syringe pump or reciprocating pump with high constant flow rate. And last one is detectors, like concentration sensitive detectors, bulk property detectors, refractive index detector, that is called RI detector, and many more detectors are used. So let's see the applications of GFC. So in protein purification, we use this technique. In DNA and RNA isolation, in polymer characterization. So polymer is a compound. It is not only used as sustained release coating agent, but also it is used in packaging system or even in our pharmaceutical manufacturing area. In the wall, floor, we also used polymer. So polymer has a diverse role from the excipients in pharmaceutical dosage form to the pharmaceutical packaging system. So in polymer characterization, we use this GFC. The last one is cell culture media and like agarose. Okay, these are the different cell culture media for quantification or the analysis of this. We use gel filtration chromatography. So these are all from gel filtration chromatography. If you have any confusion, please inform me in the comment section. Thank you.